Could y'all close the blinds back there so you can see a little better at the screen? So gene expression basically is the DNA making protein. And then when it does that, you get the traits that you see. Gene expression is regulated carefully, both in prokaryotic cells, which are bacteria, and eukaryotic cells, which would be everything else, plants, animals, humans, fungus, yeast, all those things would be eukaryotic cells. So both of those kinds of cells have DNA. The DNA makes protein, which is how those genes are expressed. And in both cases, the cells have to regulate the kinds of genes that are expressed. So every cell is different, especially cells that are in your body that are specialized. You have heart cells, brain cells, bone cells. They all have different traits, which means they have different genes that are turned on and turned off. Controlling the expression of those genes makes them have different functions and different properties. So by being in a certain part of the body, there's different chemicals which can then take those genes and make them either turn on or turn off. And that creates different cells you have in your body. So look first at the simplest cells, the prokaryotic cells. Prokaryotic cells can turn genes on and off by controlling transcription. So it just goes to the source. When DNA makes mRNA, it just controls that and has it either turn on transcription and go forward or turn it off and stop transcription, which then stops the gene from being expressed. So there's different parts to it that turns the gene on and off. One part is the promoter. That's a DNA segment that allows a gene to be transcribed. Basically, it tells the body or the cell where to start transcription. And that DNA segment with the promoter is basically the starting part of the DNA. And it tells the cell where that gene starts. Then you have after the promoter an operator, which is basically a switch that turns it on and off. It turns the gene on or off. So we see these operons in prokaryotic cells. You have the promoter, you have an operator, and then you have at least one, sometimes more genes that code for the proteins needed to do a certain job. So we don't see operons in eukaryotic cells, they're more complex, but we do see operons in simple prokaryotic cells like bacteria. So we're going to look first at that, then we'll move on to more complex cells. So operons are most common in prokaryotes and not very common in eukaryotes. And they found different operons that do different jobs within bacterial cells. The first operon that they discovered is called the lac operon. This was not even just the first operon, but the first way they discovered how genes are regulated within a cell. Lac is short for lactose. And this is a set of genes that controls how lactose is broken down within cells. And it's only for bacterial cells. Our body breaks down lactose in a different way than we're going to see here. But this is how bacteria break down lactose. Yes. So they found within a bacterial cell that the lac operon has 3 genes. And these 3 genes code for enzymes that break down lactose. They work together to make enzymes that can digest lactose. what they discovered too is that it only is expressed. These genes are only turned on when there is lactose present in the cell. When the cell has lactose to break down, these genes are turned on. But if there's no lactose present, the genes are turned off. And that amazes scientists. How does a cell know to turn off or turn on genes based on lactose being present? And it turns on those genes that code for lactose digestion. And so if there is lactose present, the genes are turned on, it makes a protein that digests lactose. If there's no lactose present, those genes are not working. And that helps save resources in the cell. It takes energy, it takes nutrients to make these enzymes. And the cell can conserve energy, it can conserve resources by only turning on these genes and making enzymes when it actually has lactose to break down. So we're going to learn today how it does that and how it turns on and turns off the genes based on lactose being present in the bacteria. So everybody these down? Okay. So bacteria can feed on lactose. Basically lactose is milk sugar, and so it's one kind of sugar that bacteria can eat. And that's how we get yogurt and cheeses. You add different kinds of bacteria to milk. The bacteria breaks down the milk, and it creates different textures, different tastes, and that gives different kinds of cheeses. So one kind of bacteria gives you mozzarella cheese, different kinds of bacteria gives you cheddar cheese, Colby Jack. So a certain kind of bacteria added to milk gives you cheese or gives you yogurt, gives you different varieties of cheese and yogurt. And then other things like kefir is a combination of bacteria and yeast. And that would be, so additional microorganisms that eat the milk. And that produces other things like alcohol and other products besides just the milk product that you get. But the organisms working on the milk will change the milk, break it down into different things, whether it's cottage cheese, harder cheeses, softer cheeses, different tastes. That all depends upon the bacteria present. All of these done? Okay. So this is what the lac operon looks like in the bacteria. So basically it's a switch on the DNA strand that turns genes on and off. So the lac operon is turned off when lactose is not present in the cell. And that's what the top picture shows you. So with that lactose present, what happens is you have this repressor protein that is stuck to the operator. And it blocks RNA polymerase from going down the DNA strand. And as long as RNA polymerase can't go down the strand, you can't express the genes, and you don't have genes being expressed, and so the genes are turned off as long as you have a repressor right here.
So it just senses the lactose better?
Yeah, so it doesn't actually sense it, but what happens is that when you have lactose in the cell, this is blue ball right here shows lactose, it will bind the repressor right here, change its shape, and knock it off the gene.
So what ferments it underneath, or what comes on top?
I mean, either way, once it hits it, it will stick to it, and it will change its shape, and it will just fall off at that point. And so with lactose being present, it doesn't actually have a brain, it doesn't sense lactose, but the lactose will hit it and will change its shape, and it will fall off, and that will then turn it on at that point. So RNA polymerase up here is blocked. It can't go down the DNA strand and transcribe these genes, but with lactose present, lactose binds to the repressor and it knocks it off. And now it can go down the DNA strand, make RNA, which will make proteins, which will then turn the genes on. So the lac operon is turned on when lactose is present because lactose can then hit the repressor protein right here and knock it off.
Is that why it takes so long for people to completely metabolize anything? Because the less of it you have, the less times it'll run into operators?
So operons, like we're seeing right here, is more for bacteria than for humans. So we don't see this as much in human cells. It's a lot more complex in human cells. Um, but still, we have reserves of enzymes in the pancreas and the liver and the intestines and the stomach. Our body makes up enzymes and stores them, and so they're ready to use. So that's really what's happening. Now, why does it take a long time to break down certain foods? Well, it's just because that it just has a lot of energy required to expend to do that. And the body is over time making more enzymes if they're depleted, but it's not the food present that will turn on the genes. It's the enzyme levels being depleted that will turn the genes on.
For example, that's interesting about the enzyme levels, but for example, if I eat lactose, right, I'll have the energy of the bread for like a few hours with the fully, or not lactose. If I eat gluten, I'll have the energy of the bread for a few hours, but it'll take me like 2-3 months to fully metabolize and get it out of my system.
2 or 3 months. Who told you that?
Apparently a not very credible source.
2 or 3 months.
Well, to like completely remove any lactose or gluten from the system.
That's interesting. Huh.
I don't know about the time frame. I just kind of eyeballed it, but it was definitely around like 1 month, 1.5 month time frame. Or for example, I know with certain drugs like I know marijuana, when you smoke it, it's active for a certain amount of time, but even for like 3 months after.
Yeah, it can stay in your, that's right. If you're a heavy marijuana user,
Yeah.
yes, it will stay in your body for months afterwards. That's true. Yeah.
Even if it's not heavy, it'll linger.
It can linger for a few days usually. Not usually more. If you're a chronic user, you use it every day, you use it heavily,
Yeah,
and you just stop one day.
yeah.
Yeah, it'll be in your body for a few months afterwards.
Yeah, so it's like
Yeah.
you
So
don't
that's
feel the
it's in your
effects anymore.
bloodstream. And so I could see gluten traces maybe in your bloodstream. I thought you meant like your intestine in your
No,
stomach.
no,
Yeah.
I'm saying.
But like traces in your blood, I could see, yeah, that could make sense to be there
There's
for.
so little of it, maybe your body doesn't detect it as often,
Yeah.
or it doesn't attack it.
No, that would make sense. You'd have some left over in your body for, I just misunderstood what you were saying, but that does make sense.
Yeah.
You'd still have some traces, very, very small amounts present in your blood for a while. That would make sense to me too. Yeah, so if you have a certain allergy or sensitivity to gluten and you eat some, then yeah, that might affect you for longer than just a day. It might affect you for a week or two, or maybe even longer than that. That is definitely true. I could see that happening. So because, yeah, so there are some things that stay in your body for a while. Basically, your body can store energy. It stores things like gluten is a starch, and it can store starches for a long time in your cells. Basically, it starch is a long chain of sugar molecules that can store for a while. So that was prokaryotes. Now we move on to eukaryotes, which is more complex. So eukaryotes regulate gene expression, but it's a more complex way to regulate gene expression. So this is done with using several different chemicals. You can see right here, it's on a straight line. Within a eukaryotic cell, the DNA is looped around. It's not just straight. You have several twists and turns to it. And that can make certain genes turn on, turn off based on the loops that you see. So different sets of genes are expressed in different types of cells. And that means, a brain cell versus a muscle cell versus a skin cell. Within the body, each individual cell has different genes turned on and turned off to make those cells work differently. Transcription is controlled by regulatory DNA sequences and protein transcription factors. So you have within the DNA sequences, stop and start sequences. So the TATA box right here, that is, let me go back for a second and see. That is a 7-letter nucleotide sequence, and it's TATAAA. And that nucleotide sequence tells it to start transcription. And so that's found in lots of genes in your body. It's the beginning start to transcription. You also have chemicals, you can see right here, that can link between the DNA strands. So this enhancer can link from this part to this part. So it links from far away strands. You writing down your notes, Dominic? Did you get those down? All right. Put the balloon down for now, please. So you can, thank you. So enhancers are ones that will reach across the DNA strand. Actually, let me see. Sorry. Did you get these? Yeah, at least does this. Did you get those down, everybody?
Yes.
Let me see.
So the AUG on mRNA, that signals the start of a, wait,
So
what's
the AUG would be the start of translation. That would be on the mRNA strand, not on the DNA strand. So TATA is on the DNA strand.
Is that at the beginning of the gene or is that at the beginning?
It not always be in the gene, no, because you can actually in this case the gene's up here. So beginning of transcription, but transcription makes the mRNA, but the mRNA is then processed and cut out different sections. So it would transcribe this area right here, but AUG would not be up until here.
Oh,
So there's parts that are transcribed that just are not used for anything. And that's what would happen in this case.
so then how would you start transcribing the gene? Is that regular CATA, the gene?
So it's not actually in the gene, but it tells you to start transcription. But it would start transcribing right here and going all the way up to here. The mRNA strain would then be all of this that was transcribed, but then it's going to be processed where it would then cut out from here to here and only have from here to here. Then you have a start codon right here where that would start the gene. Okay, so most eukaryotes have a TATA box promoter, which again is just a 7-nucleotide sequence where it's TAT and then 4 A's after it. And that's just a start signal for transcription. And then there are enhancers, which are proteins. You can see it right here, an enhancer. It links up with different parts of the DNA strand, and that helps the RNA polymerase know where to start, and it pulls it along. It makes it go faster. And then there are silencers too, which just do the opposite. They interfere with the RNA polymerase. They block it. So a silencer would be over on this side. It would block the gene from being expressed. And so if there are certain genes you don't want to express, then you have a silencer blocking the gene. So that's one way to control gene expression. Also, how tightly wound up are the DNA strands. So certain cells have DNA strands tightly wound up in certain places, which keep the genes from being expressed. Other parts of the gene are loosely bound up, and that then allows them to be transcribed more easily. And so by controlling how tightly bound up the genes are, you are controlling the rate of transcription.
Is that why like in a chromosome, the telomeres like really indexed is really close together?
Yeah, and also for protection too, because you want the telomeres to be really, really tough, because that's the part that is exposed and vulnerable to damage.
I meant like the centromere, so the middle part.
Oh, the centromeres.
Tightly condensed, because it gets smaller in the middle. It's not because it's tightly condensed, it's just less there.
Exactly, it's pinched right there. Basically, it's like wearing a belt or rubber band. It pinches that
There
area.
wouldn't be a there, or-
No, there wouldn't be.
So is that why it's so tightly expressed?
Mhm. Each gene has a unique combination of regulatory sequences. And so each gene on its own has certain start codes and stop codes to tell it when to express and when to stop expressing. So we would not call this an operon. An operon is in bacteria and prokaryotes. This is not, it's similar to an operon, but it's more complex than an operon. So we do not call this an operon. We see operons mostly just in eukaryote and prokaryotes. Okay, after it's transcribed, we get RNA. And so then RNA is processed before going on to the ribosomes. RNA processing is an important part of gene expression and regulation. So before it is able to be used to make the protein, there are parts of the RNA that are not useful, that are not used to make the protein. They have to be cut out. So the introns are not useful to the ribosomes. Intron for interfering, they interfere with the expression, and exon for expressed. And so the exon parts are the expressed parts of the RNA. They're used to make the protein. The introns are the interfering parts of the DNA, which are not used for gene expression. And those are really, as far as we know, aren't used for anything. They're called junk DNA because we don't know what their purpose is. They probably do have some purpose. We just don't know what their purpose is exactly.
Do they have nucleotides in them?
They do have nucleotides.
So how does the processor know what is needed and whatnot?
And that's a good question, but there's different proteins that will read the nucleotide sequence and know what parts are important and useful for the gene. It recognizes the gene. It knows a gene is there and what the gene should read, and it knows what parts are not part of the gene and cuts out those parts. So there are a few theories about why we have junk DNA. One theory is that by copying more of the strand, you have a greater chance of copying a part incorrectly that's an intron. So with mutations, if you make a mistake in the intron, that's not going to hurt the body because introns are not useful for anything. If you make a mistake in the exon, that's different. That is a mistake that's going to harm the body. So possibly it's used to protect you from mutations. You just have a greater length of DNA. You have a greater chance of, if you do have a mistake, making the mistake in the intron section. So that's one theory about why we have introns. Another theory is that it gives you more possible combinations of expression of genes. So if you have these parts to it, you have these three parts, you could move them around and mix and match different parts, which possibly could cause you to have more protein variety. And so it's possible the cell is able to then take these exons, rearrange them to create a wider variety of proteins. And you can have fewer genes and get more variety of proteins from that one gene. So those are two reasons why we might have this, but we still don't know for sure why we have it. There's three parts to mRNA processing. You have introns removed. So introns are non-coding regions. They interfere. And those parts are then taken out. Then you have exons, which are the coding parts, which are spliced together after the introns are removed. Then after that's done, the last step would be to adding a cap and a tail. So it's just more nucleotides that are added on at the front and at the end. So the cap allows it to bind to the ribosome. So mRNA is going to go to the ribosome at this point. So the cap allows it to do that. The tail gives it protection as it leaves the nucleus and it has to go through a small hole. It protects it from being damaged because our mRNA is very vulnerable to damage. And so it just protects it as it hits the wall of the nucleus. It hits the tail part.
So is it the same thing as like telomeres?
Exactly right. Same exact thing as telomeres. Yes. So those are the three steps. Did you actually, sorry, let me go back for a second. Did you get this done? We got this, okay. Okay, so a cap, as I mentioned, it's a nucleotide that is added to the beginning of the mRNA strand. And that helps mRNA bind to the ribosome. So after it leaves the nucleus, it will go to the ribosome. It helps it stick to the ribosome. Sorry. There we go. And so it's just one nucleotide. But the tail usually is more than one nucleotide. So mRNA is even more vulnerable than DNA to damage. DNA is double helix, double stranded. It's more stable. RNA is less stable, and it's also only one strand. And so it is going to fall apart more easily than DNA. And that's why it's important to have a cap and a tail on it as it travels through the nucleus to the ribosome.
So where does processing happen? Is it?
It's in the nucleus.
Is there like a certain thing that it goes through?
Yeah, the nucleolus. So, the nucleus contains DNA and protein. And so after it transcribes in the DNA strand, it leaves the DNA strand, but it's still in the nucleus, and there's proteins around it that will then splice it and then bring it together to process it. And then a tail is a string of nucleotides at the end of the mRNA strand. And that just gives it protection as it leaves the nucleus. Sorry, let me go back to that. There we go. So it's a very small hole it has to go through when it leaves the nucleus to the cytoplasm where the ribosomes are. And it will wiggle through their tail first. So the idea is that only the tail is affected and not the rest of the mRNA strand. So the vast majority of DNA, we don't really know its purpose. It might do nothing, or it might do things we don't know about. But we have discovered about 30,000 genes or so in the human DNA strand. But there's billions of other nucleotides. We don't really know what they do. And it might be that they do nothing, or just we don't know. But the majority of the DNA strand we don't know anything about. So a lot more genes to be discovered than what we know about so far. Okay, so I want you today to work on page 237 and do questions 1 through 5 today. Tomorrow we'll do a lab, and then we'll start to review on Friday and Monday for the test.
Wait, Monday's the test?
No, Tuesday's a test. So tomorrow's a lab, and then Friday we'll do the chapter review. Monday we'll review, and then Tuesday will be your test.
Oh, I'm sorry. I must have forgotten which one.
Oh, okay.
This is the 18th.
Okay, I'll give that 1 second.
And also, do I need